Good afternoon. Today we're going to talk about Chapter 3, which in our textbook corresponds to the chapter about cells. And in terms of the chapter on cells, what we really want to focus on is the plasma membrane, the movement of substances into and out of a cell, because from an anatomy standpoint, we want to work at sort of the organ or the organ system level. And for example, when we talk about inhaling oxygen, we want to know how that oxygen gets from the air into our lungs, across that membrane, and into the tissues or into the blood. So we really want to talk about cells from an anatomical perspective. We're not going to dive deep into the cell structures, but we're going to focus more on the plasma membrane and the methods of moving substances across that membrane. All right. So the first video is looking at why this matters, why we want to talk about the body cells, and you can watch this video at another time. From an anatomical perspective, in human anatomy, our bodies are made up of different types of cells, and so from this screen here, you can see we have little round ones in terms of the erythrocytes or the red blood cells. We have these sort of purple blobby looking ones, which are going to be part of our immune system macrophages. We have the long, skinny nerve cells where you can see how electrical conductivity can happen down that extension. All different types of cells. It doesn't matter on this page what the cell does, right? There are specific characteristics of cells that all cells have. They all have a plasma membrane. They all have organelles. They all provide specific functions. Most of them will have a nucleus. So while these cells are diverse, they also have some pretty specific unifying characteristics. So a generalized cell, three things in common, basically what's called the plasma membrane, which is the outside of a cell, the cytoplasm, which if you thought about the cell as a balloon, the balloon would be the plasma membrane, the air or the water inside the balloon, if it was a water balloon, would be the cytoplasm, and the nucleus is going to contain DNA. Most of our cells have nuclei, a couple examples of cells that don't have nuclei, but most of them do, and that's where the DNA is stored. From there, the cells will have a variety of different structures based on their function. So this is a generalized animal cell. And as you can see, there's sort of that gray opaque looking plasma membrane. And then inside we have all these different colored structures. We have the green Golgi, and then we have the purple nucleus, and we have those orange mitochondria, remembering that our cells don't have all these colors really on the inside of them. This is just for effect. But each of these different organelles provide a different function. And we're going to focus really on the plasma membrane and the nucleus. So when we talk about cells inside the body, of course, cells work together to make up a tissue. And so the cells themselves are just one part of a tissue from an A&P perspective. So we have the cells, and we have other things. We have extracellular fluid, which is interstitial fluid, plasma, cerebrospinal fluid, any kind of body fluid in the body. We have cellular secretions. Some of our cells produce things, saliva in the mouth, mucus. And then we have what's called the extracellular matrix, and there's sort of a sticky substance that holds the cells together, has some fibers inside. These are extracellular, outside the cell materials in the body that help to make up a whole organism. Also remember that these are not just human anatomical structures. Extracellular materials are found in plants and animals and all sorts of other organisms. All right, so the plasma membrane. So you'll notice in the image on the right that you'll see a little square with an arrow, and it's showing you what sort of an expanded view of the plasma membrane would look like. You can see on the right the little white spheres with two tails extending from them. Those are called phospholipids. Anything that's purple is going to be a membrane protein, and anything in the green hexagons that are sticking up off of those proteins is going to be called surface sugars, called the glycocalyx. And each of those provide specific functions to create a functional plasma membrane. And again, We have the phospholipids, we have embedded cholesterol, we have membrane proteins, and we have carbohydrates. And again, all of those are going to provide different functions. The phospholipids provide that barrier. You'll notice the inside of the cell is that orange color. The outside of the cell is the whitish green color. And substances have to move between those two sides. We want to bring in sugar and oxygen. We want to get rid of carbon dioxide, cellular waste, and things like that. They have to cross that membrane. A variety of different ways that we're going to get to as to how they do that. Membrane proteins, those can do everything from join cells together to enzymes to the one on the left that looks like it has a little tunnel going through it. That's where substances can pass through from one side of the membrane to the other. So again, each of these structures have a specific function. You can take a look at this animation on membrane structure, be super helpful. The lipids that we spoke of, those phospholipids, which have the round spherical polar phosphate head end, and then the two fatty acid tails, right? 75% of those. A couple of them are what are called glycolipids, and then 20% is cholesterol. So within the membrane itself, we have these cholesterol molecules. And we often think about cholesterol as being a negative thing. And it is a negative thing when it causes clogging of your arteries, etc. But the body needs cholesterol. The body makes its own cholesterol. We don't have to take it in. But the body needs cholesterol for membrane stability. What cholesterol does is it helps to hold the membrane together when it gets too hot, and it helps to keep the membrane flexible when it gets really cold and icy. So cholesterol does have its place. Nope. Transport proteins. This is going to be a little animation that you can take a look at that will give you a little bit more information on the role of transport proteins. Now, with these Panopto videos, I'm not exactly sure if you can view an embedded video while this video is playing. So these are available on Mastering. You can go under the study area and you can find all of these different videos and animations, and I encourage you to do that. Receptor proteins is another one. Enzymes is another one. And again, when we're doing these topics, we want to go back to why is this important? Why are we worrying about this? The glycocalyx. So we have the cell, we have the plasma membrane, we have a couple of those carbohydrates sticking off. It's called the glycocalyx. This says the glycocalyx of some cancer cells can change so rapidly that the immune system cannot recognize the cell as being damaged. How about that? And a mutated cell is not destroyed by the immune system, so it's able to replicate. So cancer cells have found a way, right? The glycocalyx sticks off of the surface of our cells, and our immune system goes by and says, okay, this is something I recognize. Sometimes if one is weird or two are weird, then the immune system will recognize it and destroy that cell. But sometimes cancer cells can make it appear as though it's normal, even though these are broken. And that tricks the immune system to say, oh, it belongs here. And if it belongs here, it's allowed to replicate and reproduce. So that's a homeostatic imbalance that has to do with the plasma membrane glycocalyx. So when we're talking about the plasma membrane, right, what we wanted to do is talk about how we're getting things across the membrane. So the plasma membrane itself is what's called selectively permeable. That means certain things can pass through easily, others take more effort or energy. And so there's two essential ways. Passively, we can just have things cross the membrane with no energy. The classic example of passive transport is the oxygen into the lungs. It goes from the lungs into the alveoli, the cells of the lung tissue, and then into the blood. No energy required. All right, active transport is when we need to actively use energy to push something in a certain direction. We're going to go into a little bit more detail on this. So when we're talking about passive methods, anything that requires no energy, it usually is grouped into three types or three modes: simple diffusion, facilitated diffusion, and what's called osmosis. Now, these three things have some very specific characteristics in common. One, the term diffusion and osmosis or passive transport means that we're going to go from an area of high concentration to an area of low concentration. When I inhale, The air that I just took in has a high concentration of oxygen. The air that was in my lungs has a low concentration of oxygen because it has already been absorbed into my blood. So it's high to low, which means it's going to go from the air I just breathed in into my lung tissue, down its concentration gradient from high to low. That's going to happen whether it's simple, facilitated, or osmosis. There are a couple differences. This is an example of simple diffusion. You'll notice that there's a dye tablet in the bottom of this beaker of water. The dye tablet is a high concentration area, and the dye tablet is going to dissolve and it's going to diffuse out into the low concentration area. So by the time you get to the third beaker, it's all uniform in its color, which means it went from a high concentration with the pellet out into a lower concentration until equilibrium. That's the key to passive transport. The goal is to reach some sort of equilibrium. Most of the time in a dynamic system like the body, we're not reaching equilibrium. We want to aim for it, but we don't get there, right? We don't want to get to the point where we have enough oxygen on both sides of the membrane, because then we're not absorbing oxygen. If we don't absorb oxygen, right, we're out of luck. So the speed of diffusion is influenced by three things: the concentration, the size, and the temperature. So three things that are going to influence how fast something diffuses. All right, if there's a big difference in concentration, then it's going to fly across. Smaller molecules diffuse faster. Think about a room that's packed with people tight, tight, tight. If we wanted to get from one corner of the room to the other, it would be much easier for a little teeny tiny kid to kind of weave through to get to the other side than a big giant man to kind of push his way through. Smaller molecules diffuse faster. Something you may not have thought about is higher temperature. The random movement of molecules as they diffuse, right, is sort of their inherent kinetic energy. The warmer something is, the faster things move. The colder something is, the slower they move. So the warmer it is, the faster these things are going to move. So concentration, molecular size, and temperature. So when we have molecules or substances that passively diffuse through the membrane, usually they're going to be lipid soluble, fat soluble, and nonpolar, or very small. So small, nonpolar, and non-charged. That's going to be simple diffusion. An example is oxygen or carbon dioxide moves by simple diffusion. But say we have something that's a little bit bigger, but still we have a large concentration gradient, but it can't move right through the membrane. That's going to be facilitated diffusion. To facilitate means to help something across. So we're going to facilitate that substance across the membrane, usually through one of those proteins that we saw with the channel through it. Right? That's going to be facilitated diffusion. And then osmosis is an interesting membrane transport mechanism in that it's just the diffusion of water, but it's so special and so important to the maintenance of homeostasis in the human body and the body of most organisms that the passive transport of water gets its own special name called osmosis. All right, so a simple diffusion animation. Again, if you can't open this, please go to Mastering and take a look at it. When we talk about facilitated diffusion, we can either do a carrier, meaning something carries it across, a protein carries it across, or a channel. It can be literally a door, a tube, a hole, and the substance moves right through the channel. Some sort of protein is helping that across. Glucose, amino acids, ions are the classic examples in human anatomy. Glucose is big and it is polar. So two strikes against it as far as moving right through the membrane. It has to be carried in by insulin, or it has to be go through a channel. Amino acids can be big, they can be charged. Ions aren't necessarily large, but they definitely have a charge to them. So they will move down the gradient through a facilitated diffusion mechanism. This is a little bit more information on carrier-mediated facilitated diffusion that you can look through. And this is channel-mediated. Again, you can see on the right-hand side how there's a tube and those little red dots are going to go right through that protein, passing through the lipid bilayer without touching the membrane proteins, without touching the phospholipids themselves. It's going to go right down through. When we go through, action potentials, right? You're going to talk about the two terms at the bottom of this page: leakage channels that are always open, and gated channels that are sometimes open and sometimes closed. All right, and we're going to talk about ion channels in the mode of action potential transmission down the membrane. So that term will come up again later. Osmosis, you'll know those little, look at those little Mickey Mouse ears on the right-hand side. Those are actually water molecules. They will diffuse through the plasma membrane very slowly, right? Most of the time they're going to go through what is called an aquaporin, which is a specific water channel that allows water to go in and out or across that membrane. Now, when we talk about osmolarity, what we're talking about is how much of something is dissolved into a solvent. So if we have salt water or sugar water or proteins or ions, anything dissolved in our blood, there's going to be an osmolarity to what's going on inside the cell and outside the cell. And that's going to mean some things are going to try to move towards that equilibrium point. And so we have what's inside the cell, we have what's outside the cell, and things are going to move across that membrane to eventually try to get to an equilibrium point. Most of the time that doesn't happen, but sometimes it does. So this is a picture of membrane, right? On that, it's called a U-tube, so it's shaped like a U. It has that yellow membrane at the bottom, freely permeable. And what you'll notice is that membrane is permeable to both sugar and water. And the red dots in the first U-tube are more concentrated on the right-hand side. And with that, we have some movement until on the right-hand side of the screen, you'll see it's at equilibrium. There's an equal number of red dots on both sides of that membrane. Same volume, same number of red dots. So that's a permeable membrane to both the sugar and the water. This example, a little bit different. This membrane is permeable to water, but not permeable to the solute. Notice how the selectively permeable membrane is really, really close together now. So now those red dots can't move to the left, but we still are striving for equilibrium. So what's going to happen is the water is going to move to the right. And the equilibrium on the right-hand side, yes, both sides have an equal concentration of sugar and water, but now the volume has changed. And so from an anatomical standpoint, you can think about this in your everyday life. Most of us have eaten foods that are really, really, really salty. And so what we've done is we've taken in a lot of salt. And then what can happen is we can feel kind of bloaty. Our hands, our fingers can get a little more fluid in them. That's what's happening here. Look at that picture on the right. What happened on the right-hand side of that U-tube was there was a lot of sugar. In our case, we had a lot of salt. And if the salt can't go this way, we can't get rid of it right away. What's the salt going to do? It's going to pull water over to its side. So in this U-tube, the sugar pulled the water. If we eat a lot of, to-go food, takeout food, Chinese food, Indian food, any number of different types of foods, maybe a lot of Italian, right? We can have a lot of red sauce, a lot of salt. The salt is going to get into the body. The salt is going to pull the water into the body because we want to make that equilibrium, and then we end up swollen. All right, these are the things we want to make connections with. What are we seeing on the screen? What does this have to do with the body? So when we're talking about reaching that equilibrium and changes in concentration on both sides, there are different terms called tonicity. When something is isotonic, it means what's on the left is equal to what's on the right. When we have something that's hypertonic, it means that there is more solute than on the other side. Hypotonic means less. So if I'm looking at this picture, the one on the left, I would say that the left-hand side with the 5 red dots is hypotonic to the right-hand side, which had more red dots. The reverse would end up being true. The right-hand side is hypertonic to the left-hand side, more red dots. When we get to the right-hand side of the screen, that second U-tube at equilibrium, that's isotonic, the same number of water and sugar on both sides. Just some terminology, right, that you want to be familiar with as we move forward. So why, right? This is a red blood cell. A red blood cell in your blood or in your body prefers what's called an isotonic solution. Just the right amount of salt and sugar and water on both sides maintains that what's called a biconcave disc, happy. If we put a red blood cell into a hypertonic solution, a very salty solution, the water pulls out of the cells and into the interstitial fluid, right? Which is why we get swollen hands when we eat a lot of salt. But yet we're thirsty because our cells are thirsty. We need more water, right? When we have more water, then we will equilibrate that salt water here, and the cells will take fluid back in, and our kidneys will get rid of the salt, and we'll go back to our isotonic. Hypotonic, when you take in too much water, cells explode. They grow because the water is pouring in, and sometimes they can burst. So we don't want to have a situation where we're taking in too much water. So this is how the plasma membrane, the movement of substances across the membrane, how that plays into our study of anatomy and physiology. Again, an osmosis animation that you can go find under mastering. This is the second, so let's put passive transport over to the side. Osmosis, facilitated diffusion, all of that. That is passive transport. Now, the other methods that cells can use to pull things in and out of a cell, it's called active transport. So what if we have eaten a candy bar, for example, and our blood sugar goes up like crazy? We want blood sugar to come down. Well, what are we going to do with that sugar? We're going to take that sugar and we're going to pull it in. We're going to push it in, right? We're going to actively transport that sugar into the cells to get rid, to reduce the blood sugar. So if it's too large, if it's not lipid soluble, if it's not able to move down its gradient, sometimes we have to use energy to push things into cells or push things out of cells. It's called active transport. The classic example is the sodium-potassium pump, where it has to do with muscle contraction and action potentials, the movement of electricity down the membrane of certain cells. And we'll talk about this many more times than just today, so you can read through that. This is the Neurophysiology Resting Membrane Potential AMP Flicks video. Please go find it if you can't watch it here. You'll notice how there are positive signs and negative signs. It's talking about the electricity, the ions that we just talked about in active transport. Lastly, we have the condition, what if we want to take in or get rid of inside a cell a giant quantity of something? We're going to talk about vesicular transport. It's not really active or passive. It's different. It's a cellular eating, right? We're going to take a membrane and we're going to go around the big molecule or the big particle and pull it into the cell. It's not one molecule. It's not one ion. It's not a set of water molecules. It is a big group of things. We're going to pull it all in like a box of things. And that's called phagocytosis, bringing that in. Pinocytosis is bringing in giant amounts of extracellular fluid into the cell, right? Big, like pull and spring water bottle, pulling that in. All right, so we have pinocytosis. Again, you'll notice that gray is the plasma membrane. It's filling it up and pulling it in a vesicle. Not just one or two, right, molecules of water, whole big amounts of it. When we are pulling things in, sometimes we can have what's called receptor-mediated endocytosis. It'll only take in certain things. And so there you'll notice these receptors, right, we're binding with that green substance, whatever that is, and pulling that in. That type of receptor-mediated endocytosis only works on the molecular substance that binds to that receptor. So it can be very specific in cell types. Exocytosis, we want to get rid of a bunch of stuff. What if we have a cell that is creating a hormone or mucus or some sort of cellular waste, but we have a lot of it? We want to package it up in a membrane and push it out of the cell. Just like when we brought it in the membrane, we're pushing it out. All right, so this picture right here shows a secretory vesicle. It is going to bind to, so you'll see the membrane and the vesicle. It's going to bind to that membrane, and then you'll notice how it opens up. And at the bottom, you'll see all of those little contents released into the exterior of the cell. So endocytosis is the process of bringing in, and exocytosis is the process of exiting the cell. All right, so that's pretty much the topics that you need to be focused on for Chapter 3, the plasma membrane, the structure, and the methods of getting things across the cell. See you in Chapter 4.